Imagine a story you've been told your whole life. A foundational fact about the world is suddenly proven wrong. For over 100 years, scientists thought they knew the origin story of the world's most common freshwater fish. It was a neat tale of a single ancestor living on a supercontinent and riding the continents apart like giant rafts. But a single fossil, no bigger than your finger, has just torn that story to shreds. Found in the ancient riverbeds of dinosaur-era Canada, a 67-million-year-old fish named Acronichthys maccagnoi is forcing us to rewrite a fundamental chapter in the book of life. Its secret wasn't discovered in some grand expedition, but by peering inside its delicate bones with powerful X-rays. And what they found suggests the cradle of freshwater life wasn't a river at all, it was the ocean. Before this tiny fish swam into the picture, the evolutionary tale of a massive group of fish called the Acronichthys seemed settled. This group is a staggering success story. It includes over 500 species. We're talking about almost every carp, minnow, catfish, and tetra you can think of. They make up roughly 2/3 of all freshwater fish species on the planet, living on every continent except Antarctica. The key to their success is a unique anatomical feature, an advanced hearing system called the Weberian apparatus. The prevailing scientific narrative for over a century, known as the Pangean single freshwater origin hypothesis, was a direct attempt to explain both their unique anatomy and their worldwide distribution. It proposed that their common ancestor evolved in the vast freshwater systems of the supercontinent Pangea, likely during the Triassic or early Jurassic period, around 180 to 250 million years ago. In this scenario, the evolution of their superhearing was a direct adaptation to life in murky rivers. Then, as Pangea broke apart, these freshwater-bound fish were passively carried along on the newly forming continents. Like passengers on immense continental rafts, they drifted apart, and over millions of years of geographic isolation, they diversified into the myriad forms we see today. It was a compelling theory because it elegantly explained the global distribution of a group of fishes that are almost exclusively found in freshwater. How else could a freshwater fish from South America share a common ancestor with one from Southeast Asia without a direct land connection? The Pangean model provided the answer. However, the theory was haunted by a glaring inconsistency, a mysterious 100-million-year gap in the fossil record. While genetic data suggested an origin deep in the age of dinosaurs, the actual fossil evidence was far younger. If these fish were so successful and widespread on Pangea, where were their fossils? For decades, paleontologists had no answer. The answer, it turns out, was waiting in the rugged Badlands of Alberta, Canada, in a geological layer known as the St. Mary River Formation. This area is a rich window into the late Cretaceous period, about 67 million years ago. This was a world renowned for its dinosaur fossils, a time when titans like Tyrannosaurus rex roamed the landscape. And in the sediments of an ancient river, a team of paleontologists unearthed the unassuming skeleton of Acronichthys maccagnoi. It was just 4 to 5 centimeters long. In a land of giants, it was easy to overlook. But its true significance wasn't visible to the naked eye. The fossil was incredibly delicate, and its most important secrets were locked inside the surrounding rock. Traditional methods of chipping away the rock would have certainly destroyed the minute structures within. This is where modern technology transformed a promising find into a revolutionary one. The research team turned to a powerful, non-destructive imaging technique called synchrotron X-ray microcomputed tomography. Think of it as a superpowered hospital CT scanner for fossils. At facilities like the Canadian Light Source, they bombarded the delicate fossil with high-energy X-rays, generating thousands of cross-sectional images that were then digitally reassembled into a crisp, high-resolution 3D model. For the first time, the team could virtually dissect the fish without ever touching it. And inside, they found what they were looking for, preserved in stunning detail, a complete, fully formed Weberian apparatus. It was the oldest and most complete example ever found in North America, a smoking gun that proved these sophisticated hearing structures were already highly developed at the twilight of the age of dinosaurs. This tiny fish was about to make a very loud noise in the world of paleontology. At the heart of this entire evolutionary saga is that Weberian apparatus, a structure that represents a pinnacle of natural engineering. This intricate mechanism is a chain of 4 small, modified bones, which are actually repurposed parts of the first 4 vertebrae of the fish's spine, a brilliant example of evolutionary tinkering. Its function is to enhance hearing dramatically by physically linking the swim bladder to the inner ear. Here's how it works. A fish's swim bladder is a gas-filled sac used primarily for buoyancy control. But because it's filled with gas, it's also easily compressed by the pressure of sound waves. This causes the bladder to vibrate in response to sound, acting like a sensitive underwater microphone. The chain of Weberian ossicles forms a direct articulated bridge between that vibrating swim bladder and the fluid-filled chambers of the inner ear. The bones act like levers, amplifying the vibrations and conducting them directly to the inner ear with remarkable efficiency. The result is a staggering improvement in auditory capability. Most marine fish have a limited hearing range, typically below 200 Hz. In contrast, otophasan fishes, thanks to the Weberian apparatus, can detect sounds at much higher frequencies, often up to 3000 Hz, with some species like catfish capable of hearing frequencies as high as 15,000 Hz, rivaling the range of human hearing. This high-fidelity hearing is a game-changing advantage in freshwater environments. Rivers and lakes are often murky, limiting visibility, and can be acoustically noisy. The ability to hear a wide range of high-frequency sounds allows these fish to communicate with each other over the background noise, to detect the subtle splash of a predator or the faint movements of prey. It opened up an entirely new sensory dimension, paving the way for their explosive diversification. The discovery of this fully functional 67-million-year-old Weberian apparatus in Acronichthys was the first domino to fall. The research team then took this crucial piece of fossil evidence and combined it with a wealth of genomic data from modern fish. By integrating the anatomical data with molecular clock analyses, they constructed a new, more robust evolutionary timeline. The results of this analysis delivered a stunning blow to the long-standing Pangean hypothesis. The new timeline pushes the divergence of these fishes, the point where they split from their marine relatives, to approximately 154 million years ago, during the late Jurassic period. This is significantly later than the origin proposed by the Pangean model. Crucially, by 154 million years ago, the supercontinent Pangea was already well into the process of breaking apart. The nascent Atlantic Ocean was opening up. This new date completely changes the narrative. If the otophasan lineage arose after Pangea had begun to fragment, their common ancestor could not have lived in a single, globe-spanning freshwater system. Instead, the analysis strongly indicated that the most recent common ancestor of all these fish was a marine lineage. This leads to the most profound conclusion of the study. The transition from saltwater to freshwater was not a singular event. Instead, the evidence points to at least 2 separate, independent invasions of freshwater by these marine ancestors. One lineage went on to colonize the freshwaters of the southern continents, eventually giving rise to the modern catfishes and piranhas of South America and Africa. The other lineage independently invaded the freshwaters of the northern continents, evolving into carps and minnows. This also perfectly solves the 100-million-year fossil gap that plagued the old theory. The fossils weren't missing from Pangean rivers. They simply hadn't evolved yet. Their story began in the sea. By solving this century-old mystery, the discovery of Acronichthys has unveiled a new and equally fascinating puzzle. The new model shows that the ancestors of this supergroup were marine, but their modern descendants are overwhelmingly freshwater specialists. Most cannot tolerate saltwater. So this presents a major question. If these fish transitioned to freshwater on continents that were already separated by vast saltwater oceans, how did they manage to achieve their present-day near-global distribution. How did the ancestors of Asian carps and South American catfishes, having separately adapted to freshwater, manage to spread so widely? This new puzzle forces scientists to explore novel and complex dispersal mechanisms. One idea involves transient freshwater corridors. During periods of major climate change and fluctuating sea levels, what are now saltwater barriers might have been temporarily different. For instance, lowered sea levels could have exposed continental shelves, and massive river outflows could have created plumes of fresh or brackish water extending far out to sea, potentially forming temporary corridors. Another possibility is episodic marine tolerance. It's possible that some early freshwater otophysians retained a degree of saltwater tolerance from their marine ancestors, which has since been lost. This might have allowed certain groups to make short, island-hopping-style crossings of marine barriers before re-establishing themselves in new freshwater systems. Finally, on a more regional scale, geological events like mountain building can cause one river system to capture the flow of another. This process can instantly connect previously isolated aquatic faunas, allowing for rapid dispersal across what was once an impassable land barrier. Untangling this new biogeographical knot will be the next major chapter in this story. The story of Acronichthys maccagnoi is a powerful testament to the nature of scientific progress. It demonstrates how a single, carefully studied fossil can trigger a paradigm shift, overturning a century of accepted wisdom. The old, simple story of a single freshwater origin on Pangea has been replaced by a more complex, nuanced, and ultimately more compelling narrative. We now understand that the journey of the world's most common freshwater fishes was not a single exodus, but a series of bold invasions from the sea. Their superpower, the high-fidelity hearing granted by the Weberian apparatus, was a masterpiece of evolutionary adaptation, with its foundations laid in the ocean and its form perfected in the challenging environments of freshwater rivers. This research showcases the synergy of traditional field paleontology and cutting-edge technology. The non-destructive eye of the synchrotron scanner allowed scientists to unlock the secrets of a fossil too fragile to dissect by hand, revealing the key evidence that had been missing for so long. The legacy of Acronichthys is not just the new history it has written, but the new questions it forces us to ask. The focus of research will now shift to the great biogeographical puzzle of their global dispersal. Ultimately, the tale of this tiny prehistoric fish is a profound reminder that our planet's history is still being uncovered, and that monumental secrets can be hidden in the most unassuming of packages. It underscores that a fossil site in Canada, a lab in California, and a shared spirit of scientific curiosity can come together to reveal the deep evolutionary pathways that connect a tiny Cretaceous swimmer to the vibrant diversity that fills the world's rivers and lakes today.